Good morning, everyone. It's a pleasure to be here. Thank you for coming, and thank you to the organizers for putting a spectacular amount of effort into organizing what looks like a very nice event. And thank you for letting me, bend your ear for a few minutes about how we're hoping to use kind of AI and powerful new methods to improve the way that we do novel target discovery, and even kind of try and better understand the biologies and the mechanisms of action that we thought we understood. And the fact is, before I get going, right, it's never actually been easier. Maybe I can switch to the next slide. Yeah, it's never been easier to generate vast amounts of data in biology. We can perturb cells in countless ways, and we can measure, obviously genetic, chemical, metabolic responses to perturbations in exquisite detail. But at the same time, it's never actually been easier to lose sight of what actually matters in drug discovery. So I'll talk a little bit about that today. And because, pipeline decisions effectively are made still, at least in Novo Nordisk, by quite a small number of functional readouts, right? We spend a lot of time thinking about how we can kind of measure responses in metabolism, signaling, and cellular behavior. And these are exactly the things that are actually quite difficult still to measure at any meaningful scale. So I'll talk about how we're trying to bridge that gap to these functional assays using combinations of representation learning, systems biology, and increasingly reasoning AI-based methods, and hoping to ground these approaches back to something that can actually influence decision-making in our early pipeline. Before I get started, Novo Nordisk is a company that's focused on kind of chronic metabolic diseases, focused on type 2 diabetes and obesity. We had a little bit of a shout out to the GLP-1s in the plenum this morning. This is where we're focused. We also have a small rare blood, rare disease portfolio, but this is probably the last time that I'll speak specifically about rare disease, so I apologize for that in advance. And if we take a step back to the kind of drug discovery pipeline, effectively, traditionally, this has involved kind of mining mostly public data sets in genetics, human cohorts, publication, literature, associations with genes, diseases, and then ranking and shortlisting candidates that we take forward through in vitro testing, where we measure, as I mentioned, a bunch of these functional responses in the cells before we can engage in any kind of preclinical testing in small handfuls of candidates that we take through to animal and first human dose. And obviously, new methods in biotechnology allow us to measure ever-increasing throughput, the effect of perturbing our in vitro cells. That adds a really exciting new layer to all of this, right? And this is not new. This is something that's been going on for the last decade or so, where we can measure thousands of perturbations and responses in these cells, either to the morphology by taking pictures of them, or to the molecular state of the cells by measuring their transcriptome or proteome at a high throughput. And the question, though, then is how we integrate this in order to actually feed this pipeline. And so my role in Novo, I have a very privileged position of leading a kind of AI team whose goal is to produce methods and enable our data scientists to help move targets into the pipeline more efficiently and with higher quality and hopefully with higher probability of success. And we do that by modeling cell state, and we do it by trying to invoke as much kind of augmented reasoning as we can to support the hypothesis generation being pursued by our scientists. So if we look at what this actually means, from these high-throughput screening experiments, we can generate genome-wide perturbation maps effectively, where we knock out every gene in the genome using CRISPR, or we apply kind of panels of small molecules or biologics, peptides, antibodies, and we can measure, the morphology response through high-content imaging of these cells, and we can measure the transcriptome response, which is the molecular response to these perturbations. And we now have some pretty fancy methods that allow us to effectively encode or compress this information and learn representations in high-dimensional spaces, all the buzzwords in AI. Effectively, what we're trying to do is cluster things together, right? So we're trying to say, how does the cell morphology or the transcriptome profile of a cell respond to perturbation A versus perturbation B? And if they're close to each other in some high-dimensional space, we kind of assume that means that they're functionally similar to each other. And the field has spent a lot of time and energy in the kind of virtual cell, digital twin kind of world in building models that very, very effectively now can help us identify cell type from a transcriptome. So you give me a transcriptome, we can use these models to really, really efficiently and effectively tell you what cell type that transcriptome came from. Fantastic. What's harder is when you try and zoom in And the thing that we actually care about is modifying the state of a cell, right? We want to try and figure out if a perturbation is changing a cell to be more or less responsive to insulin, for example. That's a harder problem because you're literally zooming in to a very small section of this map of biology on the left, right? This is a slide that I stole from someone else. And it's a challenging problem, and it's also, in my view, it's not, learning these representations and learning these cell state shifts is not the end of the story, right? Because it doesn't fundamentally tell you anything that the biologist can use to progress a target, and they don't really inform the pipeline. So how we're trying to integrate these models is by effectively using these kind of encoding methods to build models that can directly predict these functional outputs. So if you're a machine learning person, we're effectively trying to build a decoder head on our encoder models that help directly predict functional readouts like mitochondrial energy expenditure and protein localization on the membrane that might mean something for how these cells are dealing with the glucose and the energy that they have relevant to type 2 diabetes and obesity. And that this scales quite well so that we can generate these kind of large foundational perturbation data sets in cell types that are relevant to Novo Nordisk. So we focus heavily on skeletal muscle, fat, liver, pancreas. We don't believe that oncology models are going to suit every kind of nuance of the biology that we really care about, and that we effectively try and build this literally a foundation on which to build these translational arms that help us inform target progression in the pipeline. And so if you boil it down to its very, very most basic, right, we're perturbing cells, we're measuring the response of those cells to those perturbations in as cheap a way as we can. Usually that's with imaging, high-content imaging, bright field imaging, and sometimes with transcriptomics, we're building then models to compress that information and predict an expensive readout, right? Something that takes the biologists a long time to measure, a lot of money to measure. And obviously this has cost implications and efficiency gains and all the things that were actually just being discussed in plenum earlier on. We can effectively, the goal here is not to replace the biologist. It's not even to replace the functional assay. It's to make sure that the genes that we're putting forward into this process are the ones that we believe have the highest probability of actually being validated in these assays to avoid wasting people's time. So it's my goal to try and make sure that the folks in the lab are actually spending more time validating targets that have a chance of being ingested into our pipeline. And we found some good efficiency gains already, and that's all well and good. But of course, these methods are still highly dependent on you generating data sets, right? And actually, it's something that I was thinking about last night. I feel a bit of a fraud speaking in a rare disease forum, but actually, I think the data limitation question is the same for you guys, right? I mean, we're dealing with low data regimes the further down the pipeline we go. We can generate lots of data up at the beginning, and then as soon as we start moving into the kind of preclinical testing space, we don't have very much data at all. So how do you deal with that? And that's something that consumes us quite a lot, is that we need to be able to kind of also support our biologists in helping build and formulate hypotheses when we don't have very much data, or maybe the assays that they're running are not as kind of directly informative as they want them to be. And so a couple of years ago, we started on this process of trying to figure out how we could use large language models to augment this clustering that I described to you from the imaging and the transcriptome high throughput screens earlier on. And the thought process with Casper Martins here, the author on this paper, was like, what's the dumbest thing we could possibly do with no data, right? If you just want to group genes together, one of the dumbest things you can do is literally just take the protein sequence and the text description from NCBI for a gene and encode those through large language models or protein language models and embed them and create the same kind of clustering maps in high dimensional spaces as we saw earlier on. And then if you're interested in what's the response of gene A or gene X here to a perturbation, but you've not measured it, if you have measured genes nearby, then you can maybe do some fancy averaging and try and figure out what the likely perturbation response of gene X is. And it turns out that even this dumbest method possible trademark was already capable of outperforming a lot of the models in the literature at the time, like the SCGPTs and the GEARS type models. If anyone's familiar with the field, these very, very expensive pre-trained foundation models based on transcriptomes. This dumb model does way better than that. which was surprising, and it kind of led us through subsequent thought processes where we're like, okay, let's try and fine-tune a large language model to understand the reasoning behind triples. So if you knock out gene A and you measure gene B and it goes up, down, or stays the same, that's a triple. Or if you knock out gene A, you want to measure the insulin response to it, does that go up, down, or stay the same? You then kind of use these fine-tuned language models to help you reason on genes that you haven't actually perturbed, and again, produce a prediction of what that response is going to be. And again, we see a meaningful improvement in predictive power when we train models to kind of start to understand these kind of relationships between the biologies that we care about. All the time, we're labeling the latent spaces either in the large language model or in the stuff I mentioned earlier on with the screening, with these labels that are the functional outputs of the cells that we actually care about for progressing targets. There's a couple of QR codes. I'm happy to talk about this afterwards if you like. And then recently, we've taken this one step further. So I'm a computational biologist by training, and I'm a big fan of systems biology, and I like the kind of probably false comfort that you get from looking at networks and pathway diagrams. But then, you know, the question becomes, how can you kind of prime a large language model so that you're not just effectively trying to say, look, with the other methods on the previous slide, we're taking a target gene and we're just trying to see what lies nearby in the embedding space. Maybe we can bring a bit more of this network understanding into this process so we can train the large language model, just like with the triples, but with, you know, more detail, basically. What are the upstream regulators and intermediate nodes and then downstream effectors of a particular perturbagen? And you might not have measured this guy, but you might have measured something else that connects the same nodes in this graph. And again, we can see actually that this mechanistic reasoning that the models then produce also produces quite a significant improvement in perturbation response. And the reason I'm talking about this is that we don't, you know, we've spent literally tens of millions of dollars generating the high throughput screening data, which we believe is a kind of proprietary asset for us to really understand how these biologies operate for cardiometabolic disease. These methods are all completely agnostic to that, right? You can run these methods yourself. They're effectively free. And they give you a reasonable readout on an unseen kind of perturbation to a cell type. So just in the interest of time, you know, we're effectively building these large foundation models to effectively encode and represent complex cardiometabolic disease biologies. We're using cheap, high throughput data to try and make predictions of more expensive functional readouts here that actually, you know, speak in the language of the biologists in the therapy areas that are our customers. And then in cases where it's hard to generate the data, or if it's, you know, if it's somewhere that's, you know, we often are interested in novel targets, novel cell types, novel regimes in biology. These are things that by definition challenge these kinds of models because you might not have gathered measurements there. How can we use kind of large language models and the embeddings that you get from them to improve the quality and the scale of the predictions that we're making? So I'm a little early, but I think I'll stop there. And if anyone has any questions, I'm very happy to kind of have a discussion about it.
Thank you. Thank you, Rob. I think you've done a great job of taking something very complicated and making it sound very simple and straightforward and clear. So that's great. Okay, questions from our audience. There are a few on this side.
Thanks for a great talk, Rob. I wonder if I could just ask a bit about the sort of decision making at the end of that sort of journey across those different pieces, because I imagine you'll have issues depending on the particular question of sort of different levels of being out of distribution, and there may be therefore different, if you like, different levels of contribution from the different sources there, whether it's the sort of text embeddings, whether it's the biology that's being embedding and the sort of inference from reasoning. Could you say a bit about how you pull that together to sort of decide actually which to sort of go after and validate, given presumably you have a large number of options and have to sort of filter that down?
Yeah, it's a really good question, and I think it comes back to what I said before, which is we're not fundamentally trying yet, at least, to remove the need to do these downstream validations. We're just, I mean, our optimization function at the moment is just the hit rate, right? We're trying to increase the rate at which we can take an experiment that would take 6 months just to, and $500,000 to profile, 200 genes, and make sure that more than 10 of those genes actually come out as hits in that experiment. That's kind of my goal at the moment, because it's just an extraordinary waste of talented people's time and energy. So the optimization at the moment is relatively easy, in the sense that we are still using the combination of literature evidence, genetics evidence, and the kind of predictions that we make in the model. Essentially, it turns into a giant spreadsheet. I think the thing that differentiates what we're doing here from oncology is that the turnaround times in oncology are fast enough that you actually maybe want to have a machine doing that rationalization loop. And folks at GSK, like Patrick Schwab will talk about this, where they have this kind of like rapid turnaround decision making. Here we have a bit more of a luxury of time, because a lot of the experiments that we do in the lab take 2 to 3 weeks. And so, in my mind, losing a day to someone making a decision with a biologist in the room isn't the end of the world. And so it's still quite a human process to tie that together. And sometimes it's a little subjective, depending on the current direction of the wind in terms of the opinions of the therapy area, right? But it's a good question, and I don't know actually if we will ever get to the point where we have proper heuristics that will allow us to do that automatically, if that's what you're asking. Yeah.
Thank you.
I'm here, Rob. So you mentioned you were a systems biologist. In a world at the moment that is quite heavily AI-dominated, data-dominated, what's the role for maybe, let's call it, maybe old-fashioned or more traditional sort of methods to bring what they can to this sort of picture, particularly perhaps in the realm of quantifying, the effects of those perturbations, rather than just sort of saying, well, this is similar to that.
Yeah. Yeah, and yes, I think there's there's the world of kind of like PK and systems pharmacology that we don't really operate in. I think for us, being able to describe why genes might be close to each other in an embedding, or why perturbation responses are the same, for me is kind of like the translation to the biologist of like the importance of this specific proximity that we've measured somewhere is the most important contribution of SysBio. You can say whether it's likely due to some kind of signaling cascade. You can say whether it's due to a protein-protein interaction, or you're knocking out multiple members. In the most obvious sense, you're knocking out 2 different complex members of the same multi-protein complex. I feel like, to me, it's kind of an explanation layer that's very helpful. It's not so much quantitative, at least in how we're using it. We're using it to try and prime the large language models to constrain and reduce the capacity for them to hallucinate completely nonsense stuff. And then we're using them as an output to try and explain to the biologist the meaning of the proximity that we've measured in one of these kind of multidimensional spaces. But again, it's a little bit more art than science than I would like at the moment, but it's something that actually the reasoning traces in the models, we're finding. We've got a paper out at, under review at NeurIPS now, and that's basically looking at the reasoning traces in the large language model. You can effectively take the chain of thought that these models give you, and you can embed that in just a kind of text embedder, and then you can ask, well, how kind of smoothly am I walking around this embedding space through that reasoning chain? And if you see big jumps, it's actually quite a strong indication that the model's just kind of lost its mind, right? And it's gone and just made a connection that's just way off base. You could do the same thing with the network and the system biology if you're bouncing around that network too much.
Yeah. Thank you, Rob. I think we have time for one very quick question, because we did start a couple of minutes late. It has to be very quick, though.
Hey, Rob. This is Javier. Thank you very much for the good conversation. I guess it's quite a surprising finding, as you say, that text embeddings work better than sort of bio-trained models. Mm-hmm. Do you think that's a property of sort of scale of how big we've made the LLMs on text, or is there a better representation of biology somehow contained in language versus the core biology itself?
I think there's 2 problems. I mean, it's a good question. I think the scale definitely helps, and I think for sure that kind of like back in the day that we did that work, the cell foundation models were all still based on, they weren't really using perturbation data so much. They were using observations of single cell type transcriptome, single cell like primary cell experiments. I think nowadays maybe you would expect them to do a little bit better because you're training these models to understand the effect of a perturbation rather than just the cell type itself. I think one problem that I have with the original kind of group bias, like guilt by association in the large language model embedding, is that you're still using these associations, right? A lot of it's coming from text. There's no real biology in the sense that there's no reason that two genes should interact with each other just because they're co-mentioned in a scientific paper. Again, this is where somewhere I think the network and the kind of cisbio injection can really help, right? Because we want to move away from biology doesn't care about associations so much, it cares about mechanism. So you're trying to ground the models in some kind of mechanistic understanding first. And I think that's also where, if you look at some of the stuff like Cell Hermes and some of the papers coming out of the Theis lab in Munich, they're starting to do this as well, right? They're starting to say, what if we prime a network, an LLM with a network? It does do better, typically. But yeah, it's a good question. We can catch up about that after.
Thank you very much. I'm sure Rob will be very happy to answer any other questions at the break. Please join me in showing our appreciation for Rob and his presentation.